Department of Biotechnology. Now we are going to protoplast culture and isolation. So, what are the content? Definitions, somatic hybridizations, cyclics, historical development, importance of protoplast, isolation of protoplast. The isolation of protoplast is divided to two mechanical, another one is enzymatic. Next, so sources of protoplast and enzyme for protoplast. First definition, protoplasts are nothing but original plant cells with lack of cell wall. But they possess plasma membrane and other cellular components. The protoplast of different species can be fused to generate hybrids. Somatic hybridization. What is the somatic hybridization? Hybridization is a phenomenon of fusions of a normal protoplast with a anucleated, that is without nucleator, and protoplasts that result in the formations of cyclids or cytoplasmic hybrids. This is the somatic hybridizations. Historical development. The term protoplast was introduced by Hansen in 1880. The first isolation of protoplast was achieved by Hellerker in 1892. 1980, a protoplast fusion to improve plant genetic material and the development of transgenic plants. Importance of protoplast. The isolation, culture and fusions of protoplast a fascinating field of plant research. The protoplast, isolations and their culture to provide millions of single cells and a varieties of studies. The protoplast in culture can be regenerated in a whole plant. It is easy to perform single cell cloning with protoplast. Isolation of protoplast. The protoplasts are isolated two techniques. First one, mechanical methods. Another one is enzymatic methods. First one is mechanical method for isolations of protoplast. First, you will take to epidermis and to plasmolysis the cells to dissections of a cells. Last one is protoplast is released. Mechanical method. The protoplast isolations by mechanical method is a crude and tedious procedure. A very small number of protoplasts, a small pieces of epidermis from a plant is selected. The cells are subjected to plasmolysis. This causes protoplast to shrink away from the cell walls. The tissue is dissected to release the protoplast. Limitations of mechanical methods. Yield of protoplast and their viability is low. It is restricted to certain tissues with vacuolated cells. This method is laborious and tedious. Enzymatic method. The enzymatic method is very widely used technique for the protoplast isolations. Enzymatic method include good yield and viable cells and minimal or no damages in the protoplast. Sources of protoplast. What are the sources used for the protoplast isolations? The protoplast can be isolated from a wide variety of tissues and organs that include leaves, root, shoot apex, fruits, embryo and microspores. Enzyme for protoplast isolations. What are all the enzymes used for the protoplast isolations? The enzyme that can be digest the cell walls are required for the protoplast isolations. Cellulose, hemicellulose, pectins. The enzymes is digested by enzymes cellulase, hemicellulase and pectinase. The protoplast isolation pH range level for 4.5 to 6.0. The temperature is maintained by 25 degrees centigrade to 30 degrees centigrade with a wide variations of incubation period that may range from half an hour to 20 minutes. So 20 hours. First we will take to young plant. The plant is leaf sterilization. We will take to leaf. The leaf sterilizations and then epidermis peeling, peeled leaf segment. Another one, plasmolysis cells. We will take to cells in enzyme mixture. What are the mixture? Enzyme mixture, cellulase, hemicellulase and pectinase. So cell wall is reductions and release the protoplast. It will be converted to protoplast and cell debris. To washing the protoplast is isolated to plating of protoplast. Next, cell wall regenerations and the clump of the cells to produce callus formation. What is the callus? So callus is the group of mass of cells. The callus is differentiations of callus tissues and will be produced plant. The protoplast of different species can be fused to generate hybrid, that is called as somatic hybridizations. Thank you.